Welcome to part 7 of this 10-part online training program on adaptive designs and clinical trial simulation. We continue the discussion of most popular types of adaptive approaches in confirmatory phase 3 trials. In the next two modules, in parts 7 and 8, the focus will shift to another popular class of adaptive designs that support adaptive decision rules aimed at treatment selection. The statistical methodology and examples will be covered in this video, and the next module, part 8, in that module we will discuss case studies and software for clinical trial simulation. The main topic of this module is a class of adaptive designs that support treatment selection at an interim assessment. And before we delve into this discussion, I think it will be very helpful to quickly go over key features of adaptive designs that I presented earlier in part 5. In that video, I introduced a class of adaptive designs where interim efficacy data could support a decision to update the trial design by enrolling more patients or increasing the target for the number of primary events if it happens to be a survival trial. The main motivation behind this adaptive approach is that the magnitude of the beneficial effect, for example, the effect size or the hazard ratio, is very commonly overestimated in clinical trials. And in general, this would also apply to general randomized studies due to optimism held by trial sponsor or by the research team. In that case, what would be the most meaningful approach? How do we face uncertainty? We used the VALOR trial in part 5. This was a phase 3 trial in patients with acute myeloid leukemia to explain that the best course of action, the most pragmatic approach, if you will be to start small, meaning that we design a trial with a smaller sample size or fewer primary events, but at the same time we predefine a rule for updating the trial's design. For example, a rule that would enable us, based on the results that will become available at the time of an interim analysis, to increase the total sample size in the trial. But that would be the case only if the interim data suggest that the initial assumptions are not realistic. They are overly optimistic. And this module deals with a different class of adaptive approaches to trial design, a different class of decision rules that we could apply at the time of an interim assessment. So what are the main goals, the main features of adaptive approaches in this class? To answer these questions, we will begin with the motivating example based on the enhanced trial that I introduced earlier in the introductory module. With this motivating example in mind, we will then define key elements of general adaptive designs that support treatment selection. And for this purpose, we will use a case study, which is based on a confirmatory phase 3 study in a pediatric population. This trial was conducted a few years ago, and I took full advantage of an adaptive approach to identifying the best dosing regimen based on an interim assessment. Beginning with this motivating example, this motivating example is more of a reminder of key principles behind an adaptive approach to selecting, to identifying the most effective treatments or dosing regimens or interventions in a clinical trial or a general study based on available interim analysis data. The enhanced trial, as you may remember from an earlier module in this online training program, this trial was conducted a little over 10 years ago. It wasn't that long ago, and yet it was most likely the first successful application of an adaptive approach to dose selection in a confirmatory setting. This trial helped put this class of adaptive trials on the map. This example shows us how much progress we have made as a community, clinical trial community, over the past 10 to perhaps 15 years in embracing and taking full advantage of innovative approaches to designing clinical trials. And to provide a bit of background information, this was a seamless phase 2, phase 3 trial, which means that this was a two-stage trial in patients with COPD or chronic obstructive pulmonary disease. Instead of designing two separate trials, the first one being a standard dose finding trial to identify safe and effective doses, and the second trial would be a confirmatory trial. So instead of using this traditional approach with two separate trials, the sponsor of the enhanced trial decided to design a single two-stage trial. So to accomplish the objectives of the separate two trials within a single trial, that trial would employ an interim analysis which would be carried out at the end of the first stage and would inform the design of the second stage of the study. And in the first stage, 4 doses of indacaterol, which was the experimental treatment, they were tested, and the main goal of the interim analysis was to apply a predefined dose selection rule and choose the most promising doses of this experimental treatment. I would like to add that the sponsor could have employed more sophisticated decision rules at this interim analysis. For example, they could have decided to potentially use the interim analysis data to support a decision to increase the sample size in the second stage, as we discussed earlier in part 5. But the main goal of this particular interim assessment was to enable efficient dose selection. 2 doses of this experimental treatment were selected at the interim analysis, and all available data from the first stage plus the second stage, all that data were used to conduct the dose control comparisons at the final analysis. This was a successful trial, and both doses that were identified at the interim analysis turned out to be effective. And looking at the trial's outcome, you could say that, with the benefit of hindsight, of course, that it would have been advantageous if the trial had included only the best 2 doses of the experimental therapy from the very beginning. Then there would be no need to worry about complex design adaptations, of course. But the information on the dose-response relationship was simply unavailable at the time when the trial was designed, and the trial sponsor was facing much uncertainty at the beginning of the trial. They didn't have much information about the dose-response relationship. After all, this was the beginning of their phase 2 stage in the development program. And instead of, designing 2 separate trials, they decided to rely on the interim data in the seamless phase 2/3 trial to identify the most promising doses of indacaterol. And the trial sponsor emphasized that the adaptive seamless design in this trial is a novel and efficient way to combine dose selection with a confirmatory stage in a single trial. Key elements and key considerations in adaptive two-stage designs that support data-driven dose selection will be introduced in this module using this case study, which is based on a recently conducted confirmatory trial in a pediatric population. This was a randomized clinical trial in infants. They were 1 to 5 months of age, and they were suffering from a complicated condition. This was infantile hemangiomas that required systemic therapy. The trial sponsor was planning to study the effects of 4 different dosing regimens versus placebo. Those were 2 doses of propranolol with 2 different treatment durations. And we will refer for simplicity to those dosing regimens as regimens 1, 2, 3, and 4. The primary efficacy endpoint in this trial was a binary endpoint, which was defined as improvement versus lack of improvement based on clinically relevant considerations. And the primary efficacy assessment in the trial was performed by comparing the improvement rates for the 4 selected regimens versus placebo. The improvement rate was defined as the rate of complete or nearly complete resolution of the target hemangioma at 6 months. And as you can see, the length of the treatment period was quite long in the study, which will lead to some potential operational challenges for this adaptive design. We will talk about this in a few minutes. Just like the ENHANCE trial, the trial assumed in this case study comprises several stages. This will be a 3-stage design. The first 2 stages correspond to a dose-finding stage with predefined rules for identifying the most effective dosing regimen for the final assessment. And then the 3rd stage, would be a confirmatory stage to evaluate the efficacy and safety profiles of the selected regimen at the final assessment, which means that 2 interim analyses are planned in this trial. So what are the objectives of the interim assessments? The first interim analysis 1, just like in part 5, where we considered a similar 3-stage design in the context of sample size re-estimation. In this case, the first interim analysis would support an early look at the efficacy profile of the selected regimens and would support potentially early stopping for futility for each individual regimen control comparison, and as well as potential terminating the entire study. And the second interim analysis would support the predefined rules for selecting the most promising regimen. By the way, just like in the original trial, in our case study, we will assume a rule that selects a single regimen for the final analysis. This will be the most effective regimen for treating hemangiomas. And I would like to add that in this module, I will mostly focus on one aspect of treatment selection rules. I will mostly talk about the efficacy comparisons, but it goes without saying that a thorough assessment of safety would need to be performed as part of the interim assessment as well. That would be a key component of a meaningful dose selection. And it would be fair to mention other adaptive options or adaptive decision rules that could have been considered or applied in the context of this case study. First of all, an efficacy stopping rule could be applied potentially at the second interim analysis. This rule would support the trial's early termination if a very strong efficacy signal is detected for any of the 4 dosing regimens. Also, a rule for sample size re-estimation could also be considered at the second interim analysis. After the most efficient as well as safe regimen is identified at this interim assessment, the trial sponsor may compute conditional power to see if the predicted probability of success is sufficiently high. And if conditional power happens to be lower than expected, this would mean that the trial is underpowered due to overly optimistic assumptions that were used in the initial sample size calculation, and therefore it would be more sensible to appropriately adjust the sample size for the selected dosing regimen, as well as placebo arm, of course. And the goal of the sample size re-estimation will be to improve the probability of success for the identified regimen at the final analysis. And we're not completely done with this slide. I would like to make one last note that a key feature of the trial design in this case study is that due to a seamless approach, this is a multi-stage study. We can think about this as a phase 2, phase 3 trial. And due to the seamless approach, all data for the selected regimen, the data from the dose finding stage as well as the confirmatory stage, will be utilized at the final assessment. And I'd like to point out that by contrast, if a traditional approach with 2 separate trials were used in this development program, then of course only the data from the second trial, the confirmatory trial, would be used at the final assessment. From this perspective, it is clear that a seamless adaptive approach is a lot more efficient than a traditional framework that would rely on 2 separate trials. For the sake of completeness, I'm going to go over additional information of generally secondary importance to define the patient enrollment parameters, as well as patient discontinuation dropout parameters and initial sample size calculations. In this trial, as you can see on this slide, the patient enrollment period is rather long in this trial. It's about 3 months long. The assumed patient discontinuation or dropout rate is 15%. As before, here we're going to assume uniform patient dropout distribution. The initial sample size calculation based on clinically relevant assumptions about the improvement rates in the individual dosing regimens that will be presented on the next slide gave us the total number of enrolled patients at 450 patients, and then accounting for the dropout rate, we end up with the 380 evaluable patients, and those are defined as patients who complete the 6-month treatment period. This slide provides information on the assumed efficacy profile of the 4 dosing regimens as well as a placebo arm. This information is presented in the table on the left-hand side of the slide. We can see here that the assumed response or improvement rate for placebo is 10%. And as we now go across the 4 regimens, the improvement rate starts at 30% and goes all the way up to 35%. What we see on the right-hand side of this slide is the definition of the effect size, which is shown in the rightmost column in this table. And this is a standard definition, which is the difference in the improvement rates for each individual regimen versus placebo, defined as pi minus p1. And it is standardized by the common standard deviation with the square root, where the pi bar is defined as the average improvement rate in the given regimen arm and placebo. I would like to point out very quickly that, as you can see, this slide defines a single treatment effect scenario. And as I've emphasized multiple times before in parts 5 and 6, for example, my general recommendation will be to compute key operating characteristics of any adaptive design, this adaptive design or any adaptive design, under multiple scenarios, treatment effect scenarios, that correspond to a more optimistic set of assumptions, and then a handful of realistic set of assumptions, if you will, that would correspond to a stronger effect and a weaker effect. And then you would select the design parameters that would result in desirable operating characteristics, for example, desirable probability of success, desirable probability of selecting the most promising dosing regimen across those treatment effect scenarios, and that will provide a foundation for a robust adaptive trial. In the next section, we'll take a closer look at key considerations for this adaptive design. This slide and the next slide are quite similar conceptually to the slides that were included in part 5 when we went over general principles for selecting the most appropriate timing for the interim looks in adaptive designs that support sample size re-estimation. And just like in part 5, we're again dealing with a 3-stage design, which is defined by 2 interim analyses, and the trial stages will be defined here using the same approach conceptually, again as in part 5. For example, stage 1 would be comprised of all patients who complete the treatment period. In this particular case, this would be a 6-month treatment period, but a cutoff date that specifies the first interim analysis. And then likewise, stages 2 and 3 would include all patients who complete the trial between the first and second interim looks, and then finally between the second interim analysis and the final analysis. And the interim data at the first analysis will be used to evaluate the need for futility stopping to support potential futility assessments, futility decisions. And we refer to it as a futility stopping rule or go-no-go decision rule. We would apply a predefined rule of this kind to each dosing regimen compared to placebo, and most commonly this rule would be defined using conditional power, and we will go over the mathematical details in a few minutes. But in very general terms, if there's little evidence or no evidence to show that each dosing regimen has a beneficial effect compared to placebo, then of course it would be most sensible to halt patient enrollment for this particular trial arm. And if all dosing regimens happen to be discontinued for futility, then the trial will be terminated at the first interim analysis, because continuing the trial in this case would be futile. And how do we choose the information fraction for this futility interim analysis? The information fraction would be defined in this case, of course, as the number of evaluable patients who complete the treatment period by this interim analysis divided by the total sample size, or the total number of evaluable patients in the trial. And whenever it comes to futility assessments, as we have said before, it is generally advisable to set the information fraction to a fairly small value, to perhaps 30% or 40%, to avoid a late interim analysis that would defeat the purpose of a futility assessment. Because if a dosing regimen truly underperforms, with an early interim look in the trial, this regimen will be discontinued as soon as possible. This would be ideal from an operational perspective. What about the timing of the second interim analysis that would support treatment selection? The arguments that are summarized on the previous slide were very similar to what I presented in the context of adaptive designs with sample size re-estimation that we discussed in part 5. Because after all, in both cases, the interim analysis results are used to decide between the go and stop decisions. And it's better to carry out a futility assessment as early as possible to save resources in case the trial needs to be stopped due to lack of efficacy. But what about the second interim look? The second interim look in adaptive designs with sample size re-estimation, as you may remember, is used for potentially updating the sample size. And in this class of adaptive designs, the interim analysis has a different objective. How does this affect the recommendation for selecting the information fraction at this interim analysis? Of course, it will be critical to schedule this interim analysis prior to closing patient enrollment. Otherwise, it will have to deal with operational issues such as reopening trial sites that were closed at the end of the patient enrollment period. But it's a bit more complex than that when we talk about adaptive designs that support treatment selection. Because a treatment selection is conceptually quite similar to a futility stopping rule. Because if a certain dose or regimen is not selected, then the corresponding trial arm will be dropped. It will be discontinued after the interim analysis. And from this perspective, it would be best to drop the doses that will not be selected for the final assessment as early in the trial as possible. And therefore, as a rule of thumb, a midpoint would be a good choice for an interim analysis that is designed to choose the most promising doses, the most promising regimens for the final assessment. For example, the time point by which 50% of the patients complete the treatment period. This will help the trial's sponsor reliably estimate the effect size and then confidently select the most effective dosing regimen, and at the same time it would save resources by reducing the number of patients that were assigned to the trial arms that will be discontinued. We will now go over key considerations for each interim assessment, and of course we will begin with interim analysis number 1. We will begin with a set of assumptions. As we argued a few slides back, it would be best for the first interim analysis, because it is designed to support futility assessments, to set the information fraction to a relatively small value. In the context of this case study, we're going to set the information fraction for the first interim analysis to 20%. This will be done mostly to reduce the number of pipeline patients, and 20% of the evaluable patients, which will be a total of 75 patients, will then complete the 6-month treatment period by this first interim analysis. And what about the pipeline patients? The pipeline patients, we have talked about the role of pipeline patients in general adaptive designs. In this case, they would be defined as patients who are enrolled in the trial prior to this interim analysis, but they do not complete the treatment period, which means that they are not evaluable, and therefore the outcomes from this set of patients will not be used in the futility assessment. As we have done before, for example, in part 5, in the context of adaptive designs with sample size re-estimation, it is always very helpful to examine the impact of potentially fairly fast patient enrollment on main characteristics of an adaptive design. And as a step in that direction, we're going to take a look at a figure which is conceptually quite similar to what we discussed back in part 5. In this case, we see a plot of the number of enrolled patients, represented here by the yellow curve, and the number of evaluable patients in the trial. This is the red curve. As a function of time, it is measured on the horizontal axis in months. And as we said just a minute ago, the interim analysis, the first interim analysis, will be held after the 75th patient completes the 6-month treatment period. This number of patients is represented by the horizontal dashed line, and to be able to find the milestone in months from the beginning of the study that corresponds to the first interim analysis, we need to follow this horizontal line until it crosses the red curve. That is expected in this particular case to happen just a little bit after 12 months. And if we now draw a vertical dashed line at this milestone and follow it until it crosses the yellow curve, we'll see that, again, in this particular case, this is just an illustration, 178 patients are expected to be enrolled in the trial by that time, which results in 103 pipeline patients. How do we get that? It's the number of enrolled patients, 178, at the interim analysis milestone, minus the number of patients who will actually contribute to the decision-making process at this milestone. And you would agree that this is a fairly large number of pipeline patients. There are actually more pipeline patients than evaluable patients at this particular interim analysis. And it should not be surprising since, as we've said before, the treatment period in this pediatric trial is quite long. It is 6 months long, which immediately implies that a large number of patients will end up being enrolled into the trial by the time the 75th patient completes the 6-month treatment period. And just for comparison, a 6-week treatment evaluation period was used in the schizophrenia trial that we considered in part 5 in the context of sample size re-estimation, and there the number of pipeline patients was much smaller. A futility stopping rule will be set up at this interim analysis for each dosing regimen in the trial. Each regimen will be compared separately to placebo, and it will be formulated in terms of the regimen-specific conditional power, which means that the decision rule for dropping a particular dosing regimen due to futility could be pre-specified as, for example, 20% or lower conditional power, which means that the likelihood of achieving statistical significance at the end of the study is quite low. And of course, as we said before, the trial will be terminated due to futility at this interim look if all regimens turn out to be ineffective and therefore they are discontinued. And we'll use a standard definition of conditional power in this futility stopping rule. This definition that you see in the slide is identical to what we used in part 5 when we talked about futility assessments for a different class of adaptive designs, but the definition of conditional power or predicted probability of success is exactly the same. And the key point here, once again, is that we make a fundamental assumption that the future data, the data after the interim analysis, are assumed to be consistent with the interim analysis results. Here is a useful visual summary. It is once again quite similar to what we presented in part 5 to help us define the concept of conditional power in more tangible terms, to help us connect the dots, so to speak, and understand how the observed effects at the interim analysis could be translated into appropriate levels of conditional power. This is the diagram that I have personally used in multiple adaptive trials where interim data were used to support the decision futility assessment. And in this particular case, what we see here is a plot that shows the response rate or the improvement rate plotted on the vertical axis, the response rate or improvement rate in the placebo arm plotted on the horizontal axis, and the individual diagonal red lines shown in the figure correspond to certain levels of conditional power. For example, this is just an example that you can think about presenting to a project team or the sponsor responsible for this clinical trial. Let us assume, for the sake of illustration, that the improvement rate in the placebo arm is set at 15%. The improvement rate in the treatment arm, given a certain regimen, is equal to 21%. By looking at the figure, we see that the corresponding horizontal and vertical dashed lines cross at a level which is very close to the diagonal line that corresponds to conditional power of about 30%. It is a helpful result, and it helps us understand how, once again, certain combinations of response rates, improvement rates that would be observed at this interim analysis correspond to certain levels of conditional power, and this is the process that facilitates the selection of a meaningful threshold for conditional power. Which takes us to the following slide that presents standard rules of thumb for selecting thresholds for conditional power to support reliable, meaningful futility assessments. The first bullet point is identical to what we discussed in part 5. We said at that time that it is common to use lower values, denoted here by C, for example, C of 10%, in general clinical trials, general randomized studies, because in this case, early termination would be triggered only if the efficacy signal is very weak. However, there may be some exceptions, and in clinical trials that are conducted for life-threatening conditions, it may be more clinically meaningful to consider higher values for this threshold that could be perhaps as high as 30% that will support a more aggressive quote-unquote rule for futility assessments. And in addition to that, a mathematically optimal approach could be potentially considered if it is relevant in the context of a clinical trial or a general research study, and you can find out more about the underlying methodology and its implementation in the technical manual for the futility rule function, which is included in the Mediana Designer package. But before we wrap this up, I would like to point out that, of course, these are general recommendations, and it will be very helpful to run clinical trial simulations to help calibrate key parameters of this adaptive design, which includes the threshold for conditional power. Because if the probability of early stopping for futility exceeds 10%, you may want to go back and adjust the threshold to lower the probability of early stopping for futility. What we need to remember is that with a high probability of futility stopping, the probability of success in the trial, the probability of overall success in the trial or statistical power will be reduced, and this is why it is important to ensure that the probability of futility stopping is not too high. We will go over those considerations in much more detail in the next module in part 8, where we get to talk about clinical trial simulation exercises for adaptive designs that support data-driven treatment selection. And also, we're now ready for a discussion of key considerations for the second interim analysis. The main goal of the second interim look is to identify the best regimen that will be examined at the final analysis, which means that we need to predefine those selection rules, or sometimes they're known as graduation rules, in a seamless phase 2, phase 3 trial that would be similar, for example, to the ENHANCE trial that we use as a motivating example earlier in this module. A fairly large number of candidate dose levels are typically studied in a phase 2 trial in the dose finding stage, and then the best doses are selected or graduate, quote unquote, to the confirmatory stage of the trial, and this is where this terminology comes from. Using an approach similar to what we presented just a few minutes ago, let's take a look at the relationship between the anticipated number of enrolled patients and the number of evaluable patients at this second interim look. Once again, the number of patients in this figure is plotted on the vertical axis, and the time from the start of the study is presented on the horizontal axis. And therefore, if this interim analysis is held after 190th patient completes the 6-month treatment period, the interim analysis will be held after the 190th patient completes the 6-month treatment period. And looking at this figure, we'll see that in this particular case, this is expected to occur about 23 months from the beginning of this study. A fairly large number of patients will be enrolled into the trial by that point. The figure shows that this number will exceed 300 patients, but the important point here, as we emphasized before, the second interim look will be held before the end of the patient enrollment period, and therefore there will be no need to worry about potentially important operational issues related to reopening the trial sites. We will now go over popular rules for dose selection in adaptive trials. The rules are defined on this slide, and I will jump ahead a little bit and say that any rule for selecting the best doses or dosing regimens or interventions could be used in an adaptive trial as long as it is prospectively defined. Because remember, we have said this multiple times before, that design adaptations in the context of confirmatory phase 3 trials must be pre-planned. Plus, the decision rules are not binding, and then they can be potentially modified at the time of the interim assessment. Over the next several slides, I'm going to illustrate the following rules that are quite popular in the context of adaptive designs for data-driven treatment selection. The first one pre-specifies the number of best performing treatments. For example, at the beginning of the study, the sponsor might prospectively state that 2 best doses or 2 best regimens will always be selected at the interim analysis. Rule number 2 reformulates this approach based on the effect size. In this case, all treatments with a large enough effect size, for example, effect size is greater than a clinically relevant threshold, will be selected. Rule number 3 could be applied if the treatments are ordered. For example, they represent increasing dose levels over an experimental treatments, And in this case, the sponsor may be interested in identifying the lowest dose, which is equivalent to the best dose based on a predefined equivalence bound. This will be the selection rule, and I'm going to illustrate those selection rules over the next several slides. Beginning with rule number 1, let us see how this rule could be applied to interim analysis data. What we see on this slide in the context of this particular case study is this figure that shows us the interim effect sizes for each individual regimen. They range from 0.23 all the way up to 0.43. And let us assume that in the context of this rule number 1, the sponsor would like to identify the single best regimen. This would be the regimen that corresponds to the highlighted bar on the right, the regimen corresponding to the largest effect size. This will be the only regimen to be selected at the interim analysis. Alternatively, if rule number 2 is clinically relevant in the context of a certain clinical trial, the trial sponsor may predefine the threshold for identifying the regimens that are promising in terms of the effect size. For example, if the effect size threshold is set to 0.4, then looking at this hypothetical combination of effect sizes that are observed at the interim analysis, regimens 3 and 4 will be selected because their interim effect sizes exceed the threshold. It will also be helpful to discuss the relationship between futility assessments and treatment selection. In this case, assuming rule number 2, we're going to suppose that regimens 1 and 2 are dropped at the interim analysis because their effect size were quite small. They did not look very promising at that time. And in that case, regimens 3 and 4 will both be selected at the second interim analysis if the best two treatments are to be chosen. However, if it turns out that the first 3 regimens were dropped at the first interim analysis and the sponsor is still interested in applying a treatment selection rule that always identifies the 2 best treatments, that rule will have to be modified given the outcomes of the first interim analysis, and in this case only one regimen, regimen number 4, will be selected for the final assessment. Now that we're done with this illustration, I think it will be instructive to come back to an important feature of decision rules used in adaptive trials, because most of the design adaptation rules are not binding. This applies to futility stopping rules, and this applies as well to treatment selection rules in this class of adaptive designs. We touched upon this very important feature of decision rules for adaptive designs that support sample size re-estimation or event count re-estimation when we talked about this class of adaptive designs in part 5. And the decision rules I'm going to introduce in the last module, I'm referring here to part 9, when we get to discuss adaptive designs with population selection, will be non-binding as well. This means that these rules for design adaptation could be overridden by an external data monitoring committee, or they could be overridden by the trial sponsor without compromising the trial's scientific integrity. And why is that? We need to thank the combination function principle, which is the most popular statistical method used in adaptive trials. This principle was introduced in part 2, and at that point I emphasized that this principle works with any decision rule and it guarantees overall alpha control or overall type 1 error rate control in the trial, regardless of your choice of design adaptation rules. However, since we're in a confirmatory stage, we need to remember that in addition to mathematical principles, such as the combination function principle, there are also general scientific principles, that is important to prospectively define all elements of an adaptive design. And if there are any deviations from the predefined decision rules, it is important to provide a justification. Otherwise, the medical or scientific community will simply not accept the results of an adaptive trial. This last section deals with rules and statistical inferences that will be applied at the final analysis. Here I'm going to present a quick summary of the statistical adjustments that would need to be used for the primary efficacy analysis in the trial at the time of the final assessment. And the main concern in adaptive trials is alpha inflation. We've talked about this numerous times before, which means that the type 1 error rate or the false positive rate in the trial will be inflated compared to a traditional trial design due to data-driven decision rules. And there are typically several sources of multiplicity or several reasons why the alpha will be inflated in an adaptive setting. And specifically in adaptive trials with sample size re-estimation, alpha inflation is induced by data-driven decisions to update the sample size or update the target for the number of events. We said that we could take advantage of the so-called combination function principle to address alpha inflation in this case, and we can perform the required statistical adjustments at the final analysis. Now we're looking at a different class of adaptive designs, and this is the class of adaptive trials that support data-driven dose selection or treatment selection. In this case, there is another factor that induces multiplicity. What happens in this case is that the type 1 error rate will be inflated due to multiple treatment placebo comparisons or multiple regimen placebo comparisons. For example, in the case study that we've been discussing, in this module, there are 4 regimens compared to placebo. And to address this issue, a multiplicity adjustment, sometimes it's also known as a multiple testing procedure, will need to be applied to ensure error rate control in the trial. And multiple testing procedures are derived using the so-called closure principle that we talked about in part 2 at about the same time when I introduced the combination function principle. I would like to say that several multiple testing procedures could be used in this context, that we could potentially begin with the basic Bonferroni procedure. And the Bonferroni procedure is still surprisingly popular in clinical trials and general randomized studies, even though it happens to be the worst possible multiplicity adjustment. What I would recommend in this case is the so-called Hogberg procedure, because it will result in a more efficient adjustment. And the reason for that is because this particular multiple testing procedure accounts for the fact that the test statistics for the 4 regimen placebo comparisons are positively correlated. With this efficient multiplicity adjustment, the probability of success in a trial will not be reduced by that much, because we should not forget that any multiplicity adjustment is a penalty, and it is our responsibility as clinical trial statisticians to choose the most efficient multiplicity adjustment to reduce that multiplicity penalty, and that will ultimately help improve the overall probability of success. And if you choose a multiplicity adjustment which is not efficient, such as the Bonferroni adjustment, then your primary analysis will be penalized, and there could be potentially substantial power loss in the trial just because of that. And of course, we're considering a somewhat simplified adaptive design in this case study based on this pediatric trial. In this case study, stopping the trial early is only considered for futility reasons, not for the reasons of superior efficacy. However, if a stopping rule for superior efficacy was considered at the first or second interim analysis, this would then give us another source of multiplicity, and that source of multiplicity would also need to be accounted for in the efficacy analysis. And as I mentioned earlier, it is also fairly common to combine dose selection with sample size re-estimation. and a sample size re-estimation rule could be applied at the time of the second interim analysis. This rule then, in and of itself, would also result in alpha inflation, and it would be yet another source of multiplicity. But once again, we know how to handle this. We would do this by applying the combination function principle. So I'm using all those examples to point out that in real life adaptive trials, there will most likely be several sources of multiplicity, but please do not forget that efficient statistical methods have been proposed in the literature, they are available, and they can be employed to address all of those sources of multiplicity simultaneously and support relevant statistical adjustments in complex adaptive trials that would support reliable statistical inferences at the time of an interim analysis or the final assessment. We're almost done with this module, and on this slide as well as the next slide, I'm going to very briefly outline key ideas behind multiplicity adjustments in adaptive designs that support data-driven treatment selection. I'm going to use here a simplified version of the case study that we've used in this module, mostly to simplify the diagram. Here, as you can see in the diagram on the left-hand side of the slide, we're assuming that two regimens are compared to placebo in a three-stage trial. The first stage, at the end of the first stage, an interim analysis would be held to support futility assessments, and then at the end of the second stage, an interim analysis would be introduced to potentially identify the most promising arms. And here we're going to assume, again, just for the sake of example, that only the second regimen would be selected at the second interim analysis to support the final assessment. This is the reason why there is a gap here for regimen 2. Stage 3 data are simply not available. This particular regimen was not selected for the final analysis. How do we now compute an appropriate multiplicity adjustment? To be able to do that, first of all, we're going to compute the treatment effect p-values for each regimen placebo comparison at the final analysis. They will be denoted by p1. That's the, let's say, one-sided p-value for the comparison of regimen 1 versus placebo. And p2 would then denote a p-value for the comparison of regimen 2 to placebo. In a more general setting, when both regimens were selected for the final assessment, a predefined multiplicity adjustment would then be applied to the two p-values, p1 and p2, at the final assessment. However, in the context of an adaptive design, it is possible that a regimen could be either dropped for futility at the first interim analysis, or it could be deselected because it does not look very promising at the time of the second interim analysis. How do we then deal with the p-value definitions for the final analysis if a certain regimen is not simply available? It was discontinued earlier in the trial. In that case, the p-value corresponding to the comparison of that regimen to placebo is going to be set to 1. In this particular case, p1 will be set to 1 because regimen 2 was the only regimen chosen at the second interim analysis. And after that, we're going to apply the predefined multiple testing procedure to the resulting p-values. For example, if we were to prospectively define the Hogberg procedure as a multiplicity adjustment in this adaptive design with adaptive treatment selection, in this particular case, a significant effect would be established for regimen number 2 if its p-value p2 was less than alpha over 2. So we can see in this example that even if a certain regimen or a set of regimens or treatments is discontinued early in trial, we still have to pay a certain penalty for that at the time of the final analysis. And the main reason for that is because they were discontinued based on the available data. This was a data-driven decision that will ultimately contribute to alpha inflation. And to account for that potential alpha inflation, we need to apply a multiplicity penalty that in this particular case would be based on the Hogberg procedure. And the last very quick point is that, as I pointed out before, that with the seamless approach implemented in the trial, all data for the dosing regimen selected at the interim analysis will support the final assessment of the efficacy profile, and therefore in this case, since regimen 2 is indeed selected for the final assessment, the final p-value p2 will be based on all of the data pooled across the three stages, stage 1, stage 2, as well as stage 3. That's the main advantage of a seamless or a multi-stage adaptive design with treatment selection over a traditional approach that would have employed two or potentially more separate trials to achieve the same set of goals. This is the end of part 7. It's been a high-level overview of the statistical methodology as well as relevant operational considerations for this class of adaptive designs. And we have also set the stage for part A, which will be a review of software tools for clinical trial simulation for adaptive designs that enable data-driven treatment selection. Thank you so much for your interest in this online training course. I would like to encourage you to check out the other videos in this course and share information on this online training program with your colleagues.